Are genetically modified foods good for human consumption? This is a very hot topic and there is always a discussion that if these foods are good for us or not. In this video, I will be talking about the best approach available in the market for making genetically modified foods. And I believe it is going to be very important for us to know about GE crops. GM or GMOs or GM foods have been a very popular and hot topic of discussion, and a lot of people claim that these are good food, and there are so many people that still believe that these are not good for human consumption. So today we are going to compare those GMO food with another new technology, genome edited foods. So what are the challenges and what differences we can see? We'll talk in this video. At least we need to know where our food is coming from and how they are modified genetically in the lab. Hi, if you are new to this channel, I am Saroj Parajuli and I work in the field of genome editing and genetically modified organisms for more than last 10 years. Today, this video will focus on three major topics. First, we are going to take a look at genetically modified organisms or GM food. What are the components that are needed and how these foods are made. Second, we'll do the same thing for CRISPR edited foods. So we'll look at the CRISPR technology, what we need to make the food and how these foods are made in lab. Third, it's my favorite one. So I'm going to compare GMOs with CRISPR edited foods and see if one is better over the other. Without any delay, let's directly jump into the topic and we'll start with the first topic, that is genetically modified foods. For GMOs or GM crops, we need to find a gene fragment in another crop or another species or in bacteria and virus that will give you some kind of trait in the organism. In this video, we'll take an example of genetically modified corn because this is very popular and it's very easy to understand. So let's take an example that if there is a corn plant that has leaf blight disease. Our aim is to get rid of this leaf blight disease. And for this, the first approach we will do is we'll use GMO approach. For GMO approach, we need to find a gene that gives us resistance to this disease. Then what we need to do is we need to clone the gene from another species or another organism and then put this gene in our crop. And for this we need at least three different components. The first one is promoter, second is the CDS sequence or the gene region, and the third one is the terminator. Then what we will do is we will put all those combinations, promoter, gene and the terminator together and make a gene fragment, and then we put this one in a plasmid. When the plasmid is ready, this plasmid is being transferred into that crop with the help of some vector like bacterial vector or it could be viral vector also. One thing to remember with GM food is the gene fragment should always be present in the crop so that that particular gene is expressed in the plant and we get that disease resistant phenotype. Now, let's look the same example from another approach. We'll be talking about genome edited crops or genome edited foods. So in this approach, what we are going to do is we are going to modify the plant so that it's going to give us resistance. And for this, we don't need any resistant gene. We can directly work on the susceptible genes. So first approach is to find the susceptible gene in the crop. So after we find a susceptible gene, we need three different components, and those three components are guide RNA, Cas protein and the tracer RNA. So the guide RNA comes from that particular gene and that particular plant, and the Cas enzyme, which is a molecular scissor that cuts the DNA, that comes from another organism, bacteria. So that sequence is coming from bacteria. And the third, we need the tracer RNA, and tracer RNA identifies the Cas sequence and it will bind with the Cas sequence. So we'll use the same approach. We'll use Cas enzyme with some promoter and terminator, and also we use the fragment of guide RNA and the tracer RNA with some promoter and the terminator, and both of them can come from either virus or bacteria like in the previous approach. And now what we'll do is we similar to the previous approach, we'll make a plasmid and we'll deliver that plasmid in the plant cell either by viral vector, bacterial vector, or any other component. And one thing to remember with this technique is once the gene is integrated and it does the job of cutting the DNA, then we don't need that gene fragment. We can take the gene fragment out and we still get that particular trait. This means no foreign DNA is involved in making genome edited crops. And I'll be talking about one more thing, that is use of non-plasmid method, non-plasmid or non-vector method. So here what we can do is instead of using the gene fragment, we can use the transcript of the guide RNA, transcript of the tracer RNA, and protein of the Cas enzyme. If we make a complex and put this in plant cell, then it will do the job and it will edit the gene. We get the phenotype, and this transcript and protein will be degraded because they cannot integrate in the genome. Therefore, we get transgene free genome edited crops. This means no foreign DNA involved, no any bad DNA that we need to insert in the plant. CRISPR is that simple, no foreign DNA attached ever. Finally, let's move to the third part. In this part, we are going to compare the GM food with genome edited foods. And I will not go much in scientific detail. By this point, you might already be thinking that the CRISPR approach looks better than GMO. Yes, you are 100% right. CRISPR approach is better than the GMO approach. And I will mention 3 different reasons why CRISPR approach is better than GMO approach. There is only one thing that makes CRISPR better than GMO. That is the integration of foreign DNA. No foreign DNA will be integrated in the plant cell. So we'll get the food that is free from some kind of foreign DNA. Second is regulation and wider acceptance. So with CRISPR technology, there is no foreign DNA attached. Therefore, there is no regulation in planting those crops in the field, and there is no federal regulation for these crops. And recently, European Union has also given green signal to these genome edited foods. And the third one is no harmful expression of the proteins that we don't need. So with the GMO approach, there are some other proteins expressed in the plant that we really don't need. Therefore, the CRISPR involves no foreign DNA ever. That means there is no any other protein expressed in the cell. Our plant is free from any hormones, any enzymes, and any other foreign DNA in it. Looking at the technology, we can say that genome edited crops are far better than genetically modified organism approach that we previously used. I'm not saying that CRISPR is all safe. That is beyond the scope of this video. And when it comes to CRISPR, we need to carefully select our target because some of the genes might be involved in some other physiological effects. And if we target those genes that have other physiological effects, then it can give us severe consequences. Therefore, if we carefully select our target and don't integrate the foreign DNA, then CRISPR approach is one of the best approach available in the market. And if you want to learn more on this topic, please let me know through comments. Thank you so much, and I'll see you in next video.